Hello everyone and welcome to ITLS Academy Empower the Youth. So today in this lecture we will going to study about microbial growth part 1. So this topic is very much important in food microbiology because this topic deals with microorganisms and their growth. How microorganisms grow, how they utilize nutrients for their survival, what is their growth curve, how fast they grow. So in this particular lecture we will going to see how microorganisms grow. Now let's get started. Microbial growth. The term microbial growth refers to the growth of population or an increase in the number of cells, not to an increase in the size of an individual cell. Cell division leads to the growth of the cell in the population. So simple, microbial growth is refers to the growth of population. Growth of population of what? Growth of population means the increased number of cell. If I write here cell increase is known as growth. So this doesn't means that size of any microorganism grow. No, only the number increases. Then the cell division leads to the growth of the cell in the population. So who leads to the cell growth? Cell division. So cell divide themselves and grow into two new daughter cell. That is how the population of microbes grow. So microbes grow with the help of cell division. The parent cell divide themselves and forms two identical daughter cell. There are basically two types of asexual reproduction in microbes. So basically the cell microbes, or you can say the microorganism, grow with the help of asexual reproduction. The first one says the binary fission. So binary fission is what is binary fission? So binary fission is bacterial reproduction occurs through fission, a primitive form of cell division that does not employ a spindle fiber apparatus. A spindle fiber apparatus made of protein filaments is responsible for moving the chromosome around during the cell division, that is mitosis and meiosis, in most eukaryotic cell. Bacteria do not have these structure. The bacterial cell double in size and replicates its chromosome. Following DNA replication, the two chromosomes attach to separate site on the plasma membrane and the cell wall is laid down between them, producing a two daughter cell. So the bacteria, they grow through asexual reproduction. So bacteria grow through a asexual reproduction. So the first one is the binary fission. So in this bacteria, how bacteria grow? So bacteria here, they reproduce through fission. Here bacteria is reproducing itself through a fission. What is a fission? It is a primitive form of cell division. What is a fission? Fission is a primitive form of cell division that does not employ a spindle fiber apparatus. So it is a simple form of cell division. Simple form means, right? It is if it is a bacteria, parent bacteria, I am writing its PB, that is parent bacteria. So it divide itself through two identical daughter cell. Right? So it is a simple division where a parent is dividing itself to form a two identical daughter cell. Right? So a spindle fiber, what is a spindle fiber? So a spindle fiber apparatus, it is made up of protein filaments protein filaments that is responsible for moving the chromosome around the cell division, that is mitosis and meiosis. But it is most in the case is with the eukaryotic cell. But bacteria do not have these type of structure. They do cell division through fission, and that is a very simple one. The bacterial cell double in size and replicates its chromosome. The following DNA replication, the two chromosomes attach to separate site on the plasma membrane and the cell wall. What the cell wall is, how the cell wall do that, it lays down between them and producing a two daughter cell. Let me show you a figure to explain this. See, this is a cell wall. Sorry, this is a bacteria. Can you see? This is a bacteria. So bacteria has a cell wall, bacteria has a cell membrane, and bacteria has a elongated nucleotide. Now what does this nucleotide is doing? It is dividing the cell wall and the membrane. Can you see this? Here what nucleotide is doing? It is dividing itself and forming a two separate daughter cell. Always bacteria disintegrate in two forms, that is two daughter cell, and both the daughter cell are of identical shape, size, structure. So this is about the binary fission of bacteria. Now moving to budding. A few bacteria, some eukaryotes include yeast, may also replicate by budding, forming a bubble like growth and enlarge and separate from the parent cell. So a question may ask how bacteria divide itself or how bacteria grow. So bacteria grow through two types of asexual reproduction, that is binary fission and budding. Now you can take the screenshot of this diagram, that how binary fission of bacteria take place. Now I am moving. How fast do bacteria grow? Means how fast bacteria grow. We all know that we all are surrounded by microorganisms. Microorganisms are present in atmosphere, on the surface of your skin, on the surface of table, everywhere. Right? Microorganism can grow everywhere. It is present like in atmosphere. The most favorable condition for a microorganism to grow, or you can say to survive, is atmosphere. Apart from atmosphere, the another favorable condition is water. Microorganisms grow very fast in water. That is why our food products get easily deteriorated. Why? Because of the presence of moisture content in them. So compared to the growth rate of the most of the living things, bacteria grow very rapidly. The average, if you see the average generation time of bacteria, or you can say the doubling time of bacteria, then it takes 30 to 60 minutes for a single bacteria to grow. So you can see up to which extent a bacteria can grow. Right? So apart from this, the longest generation time is Mycobacterium leprae, that is 10 to 30 days. So a Mycobacterium leprae is a microorganism. It is a species of a bacteria that grow for the longest that has a longest generation time, that is 20 to 30 days. 10 to 30 days. Most pathogens have relatively shorter doubling time. Apart from this, Salmonella and Spectrophilus coccus aureus both cause food poisoning, and their doubling time is 20 to 30 minutes. Right? So here we are talking about two types of how fast do bacteria grow. Like they can double their generation time, like they can grow in 30 minutes or in a 1 hour. Right? So you can see which extent bacteria is dividing itself. Right? So the longest time is 1 month. Then apart from this, a Salmonella is a species that it is a species that causes most of the time fever. So they grow in a very less time, like 20 to 30 minutes. In 20 to 30 minutes they can easily grow themselves. Now let's talk about the growth curve. Now coming to the growth curve. So since bacteria are easy to grow in labs, their growth has been studied extensively. It has been determined that in a closed system or a batch culture, no food added, no waste removed, bacteria will grow in a predictable pattern, resulting in the growth curve composed of four distinct phases of growth. That is, the first one is the lag phase, second is log phase or exponential phase, third is a stationary phase, and last is death or decline phase. So since bacteria can easily grow in labs, so their growth cycle can easily be studied, right? And this case is only with the bacteria their growth is so extensively studied. So it is determined that whenever it is a closed system or a batch culture, means closed system in sense that no food is added, no waste is removed, everything is closed, एक बार गया, तो that it is a closed system. So bacteria will grow in a predictable pattern in the sense that resulting in a growth curve that is composed of the four phases: the lag, log, stationary, and death. Additionally, this growth curve can yield generation time for a particular organism, that is the amount of time it takes for the population to double. So growth curve can also lead that how the bacteria is growing, means what is the how the population is doubling, what is the amount of time that is taken by a bacteria to double its population. So simple, if I draw a growth curve, then this growth curve look like this, means if I look here, that it's a bacteria, number of bacteria, it's a time. So can you see, this phase is known as lag phase, this is log phase, this is stationary, and this one is the death phase. So I am marking every growth, every phase with different color. So the green color says the lag phase, blue color says the growth phase, orange is stationary, and the last one, that is death, is with red color. So this is the number of bacteria and this is the time that is needed. So in the presence of a fresh growth medium, bacteria show the following four phases during their growth. The first one is the lag phase, second is log, third is stationary, and last one is the death phase. So in the lag phase, during this phase, the green one, bacteria here is preparing for growth, preparing for the division. So before dividing themselves, they utilize the nutrient that is present. So they prepare themselves, and the lag phase is also known as the preparative phase, where the bacteria is preparing themselves for the division and they are utilizing the nutrient in this phase. Then comes the log phase. So the log phase is that phase where the bacteria has utilized all the nutrients and here the bacteria is dividing themselves. Then comes the stationary phase. Stationary phase is the phase where the number of nutrient is equal to the where the number of generation of a new bacteria is equal to the number of the death phase. Then comes the death phase, that is known as the declining phase. So let's talk about each phase in detail. So the first one is the lag phase, where the organisms are getting used to the medium and physical condition, that is they are including the necessary enzymes for the growth. So here, what is this? Here is the organisms that are getting used to the medium and physical condition, means here the organisms are utilizing all the source of nutrients and the physical conditions, and they are utilizing the enzymes that are necessary for their growth. Then comes the growth phase, or you can say that is known as the log phase. So this is a phase where the generation time is measured. So this is the phase where the generation time is measured, means this phase shows the exponential growth of the bacteria, and this is the most ideal condition for any bacteria to grow up to the maximum. So in exam they can ask you which phase is the most ideal for the bacteria to grow fast, then it is the log phase. Then comes the stationary phase. Stationary phase says during this phase the number of new cell equals to the number of the dead cell. So new cell is equal to the number of dead cell. So there is no net increase in the viable cell. So nutrients are becoming depleted in this stage, pH is changing, toxic wastes are building up, oxygen level are depleting. So in this phase, obviously there are some cells or that is dying. So here the new cell is equal to the number of the dead cell. Why? Because nutrients are becoming depleted, pH is changing, toxic waste is building up, or you can say the oxygen level are depleting. The main reason is that nutrients are depleting and oxygen level is depleting. So there is no further growth in bacteria. Then comes the death phase. So the death phase says the rate of cell death is faster than generation. Death may accelerate and becomes exponential here. So in this phase, death number of all the nutrients, oxygens have depleted in this phase, and there is no further regeneration, and the death rate becomes faster than regeneration of a new cell. So again, if we see the logarithm of a living bacteria cell, log phase says exponential increase in the number of living bacteria cell. The first one is lag phase, no increase in number of living bacteria cell, because here bacteria is utilizing all the nutrients and preparing themselves for the cell division, or you can say the growth. Second says the log phase, that is the exponential increase in number of living bacteria cells. Then comes the stationary phase, that is plateau in number of living bacteria cells, that is the rate of cell division and death are roughly equal in this phase. There is no further growth. Then comes the death phase, that is exponential decrease in the bacterial cell. Why? Because here all the nutrient, oxygen, pH, everything is changing. Oxygen and the nutrients are depleting themselves. So that is the reason no further exponential growth is possible. Only bacteria cells are dying and there is no further regeneration. So in this particular lecture, there are some questions that can be asked in the examination. So before that, let us quickly see what all we have covered so far. So we have seen what is microbial growth, what are the two types of asexual reproduction in microbes, that is binary fission and budding, how binary fission take place. We have seen this diagram of binary fission, how this nucleotide is dividing itself, and how two new separate daughter cells are formed. Then we have studied about how fast do bacteria grow, means what are the average generation or doubling time. Whenever they asked you about the average generation time, student, so you only tell this 30 to 60 minutes, unless and until they ask you about any specific microorganism or any specific bacteria, that in case what is the longest generation time, so that is 20 to 30 days, and that is Mycobacterium leprae. But unless and until they won't ask you, so don't mention this 10 to 30 days. The average time is 30 to 60 days, 60 minutes, and the longest is this. And apart from the longest, if they ask you about the shortest time, then it is 20 to 30 minutes. That is with the case with the Salmonella and Staphylococcus species, aureus. So aureus. So this is something you need to remember this. So apart from this, we have seen the growth curve. This question has maximum chances in examination, in interviews, that they can ask you about the different phases and how bacteria grow, which phase says what, right? So this is the this in this they can ask you this question. So now let's quickly let's see what are some questions. So the first question says, what are the 4 phases of bacterial growth curve? So you have to explain all the lag, log, stationary, death phase, and about them. So you can simply explain this graph by marking this and by explaining this graph. Now coming to, what does the bacterial growth curve show? Means this bacteria growth curve, what the bacteria growth curve show? So you must know the answer. I'm not discussing any answer here, so I just want you to leave the answer or any queries in the comment section below. So what are the things bacteria need to grow? Means what are the things bacteria need to grow, what are the different things like nutrients, what are the optimum pH, what is the temperature, you should know about that, about the oxygen and everything. So the question number 4 says, how is the growth measured in microbial population? So how you measure the growth of the microbial population. Then the last one says, how do eukaryotes and bacteria, that is archaebacteria, differ in their reproductive method? So you must be aware of this slide. You can take a screenshot of this slide. You can take how this like in a bacteria reproduction, that take through fission, right? But apart from this, they do the spindle fiber apparatus, so that is missing in bacteria. So bacteria do not have these structure. So how it is differ, so you can go through this slide and you can get the answer from here. So this is all about the important questions that may asked you in the interviews, that how do eukaryotes and bacteria is differ in their reproductive methods. And yes, this is all about the topic. So if you face any query, any thing you want me to repeat, or you have any queries or anything, so you can leave all the queries in the comment section below. In the next lecture, I'll be answering all your queries. And student, you please visit our website, that is www.itlsacademy.com. And this is our helpline number, that is 7080833450. You can simply call on this number and get updated of any information you want. So thank you for watching. Please do like, share and subscribe. And yes, you can press the bell icon and get updated with the new lecture that is coming up the next. And you can follow ITLS Academy on Twitter, Instagram, Facebook, WhatsApp. Thank you.